Hey everyone, and welcome. Today, we are diving deep into one of the biggest scientific breakthroughs of our time, CRISPR. This isn't just some fancy lab technique. It's a tool that's letting us, for the first time, literally rewrite the code of life itself.
So let's just start with this wild idea. What if you could open up the blueprint of a living thing, all 3 billion letters of the human genome, find a single typo, and just hit backspace and fix it? That's the power we're talking about today. It's a level of precision and control that was science fiction just a short while ago.
All right, to really get a handle on this, here's the plan. We're going to start with why we'd even want to edit genes in the first place. Then we'll look at the pretty clumsy tools that came before CRISPR. We'll uncover its totally unexpected origin story and, of course, break down exactly how it works. Then we'll explore the whole new toolkit it's created, and finally, we'll look at the incredible future it's making possible.
So, first things first. Editing the code of life. What are we really talking about here? Well, it's all about the promise of precise genetic engineering. I mean, the ability to zoom into a cell's DNA and intentionally change the sequence, to delete a faulty gene, insert a helpful one, or just tweak its function.
And why on earth would we want to do that? Well, the potential is just staggering. We could potentially cure devastating genetic disorders like sickle cell anemia or Huntington's disease. We could program bacteria to become tiny little factories, churning out biofuels or life-saving medicines. We could design crops that are more nutritious and can survive droughts and diseases, helping us feed the planet. It's a total game changer for medicine, agriculture, you name it.
Now, for years, all of this was basically a dream. Scientists knew what they wanted to do, but they just didn't have the right tool for the job. And then CRISPR came along, and it was simple, it was cheap, and it was unbelievably effective. This is the technology that took genome editing from a futuristic dream to a day-to-day reality in labs all across the globe.
But, to really appreciate just how revolutionary CRISPR is, you kind of have to see what scientists were dealing with before. So let's rewind a bit and look at the challenging, often frustrating quest for a true genetic scalpel.
Before CRISPR, you had tools like zinc finger nucleases, or ZFNs. Now, they were a big deal at the time, but honestly, they were a nightmare to design, they cost a fortune, and worst of all, they'd often make cuts in the wrong places in the genome, which is a huge problem. Then TALENs came along, and they were definitely more precise, but they were still super expensive and really complex to make. These tools showed it was possible, but they were so difficult that only a handful of labs in the world could actually use them. It created this huge bottleneck that was holding back the entire field.
And this table, this right here, tells the entire story. Just look at that bottom row for CRISPR. Where the old tools were difficult and expensive, CRISPR is easy and low cost. And where they had so-so precision, CRISPR's is through the roof. It didn't just make things a little better, it completely changed the rules of the game. It democratized gene editing, putting this incredible power into the hands of thousands of scientists. That's why it caused such a shockwave.
So you're probably wondering, where did this miracle tool come from? Some high-tech government lab, a genius inventor's garage? Nope, the real story is so much cooler. CRISPR is actually an ancient virus-fighting machine that's been hiding in plain sight, inside bacteria.
It all started way back in 1993, when scientists found these weird, repeating patterns in bacterial DNA. For more than a decade, nobody knew what they were for. They were just a scientific puzzle. But then, in 2007, a massive breakthrough. Researchers figured out it wasn't just junk DNA. It was actually a sophisticated immune system for bacteria. It still took another 5 years of incredible science to figure out exactly how it worked, but in 2012, when that final piece of the puzzle clicked into place, everything changed.
And here's how it works in nature. When a virus attacks a bacterium, the CRISPR system swoops in, grabs a little snippet of the virus's DNA, and stores it in its own genome. It's like taking a mugshot. This little snippet is called a spacer. Now, if that same type of virus ever shows up again, the bacterium uses that spacer as a guide to find the invader's DNA and then uses a protein to just shred it to pieces. It's basically a molecular search and destroy system.
Okay, so once scientists figured out that bacteria had this amazing, super precise virus hunting system, they had a brilliant thought. What if we could hijack it? What if we could give it a new mugshot, a new guide, and tell it to hunt for any piece of DNA we want? Well, that's exactly what they did. Let's break down how this incredible molecular machine actually works. It all comes down to 2 key parts. First, you've got the guide RNA. Think of this as a programmable GPS. Scientists can design this little molecule to match any DNA sequence in the entire genome. It's the search function. Then you have the Cas9 protein.
This is the heavy lifter, the molecular scissors that will do the actual cutting. But, and this is the important part, it only cuts where the guide RNA tells it to.
And the process itself is just beautiful in its simplicity. The guide RNA and the Cas9 protein join forces and start scanning along the DNA. When that guide RNA finds the exact sequence it's been programmed to find, it latches on. That's the signal. The Cas9 protein then goes to work, making a clean, precise cut right through both strands of the DNA. It's literally search, match, and cut. That's it.
Now, making the cut is just the first step. The real magic happens with what the cell does next. See, when you cut DNA, the cell's natural repair systems rush in to fix the damage, and they have two main ways of doing it. The first is a kind of quick and dirty patch job. It's fast, but it often makes tiny mistakes, which can be super useful if your goal is to simply shut down or knock out a gene. But there's a second, much more elegant option. We can give the cell a healthy piece of DNA to use as a template. The cell then uses this template to repair the break perfectly. This allows us to knock in a new sequence, either fixing a broken gene or adding a whole new one.
But here's where it gets even cooler. The true genius of the CRISPR system is how flexible it is. Scientists quickly realized it could be more than just a cut and paste tool. They've modified it to create an entire toolkit for playing with the genome in all sorts of new ways.
For example, there's something called CRISPRi for interference. For this, they use a dead version of Cas9. They basically broke its scissors. So now it can still follow the guide RNA to a specific gene, but instead of cutting, it just sits there. It physically blocks the cell's machinery from reading that gene. It's like parking a giant truck on a railroad track, turning the gene off without changing a single letter of the DNA code.
And hey, if you can turn a gene off, you can probably turn it on, right? That's CRISPR-A for activation. Here, they take that same dead Cas9 and attach a molecule that acts like a gas pedal for genes. When it binds to the start of a gene, it tells the cell, hey, make more of this. It's essentially a volume knob for the genome that lets you turn genes up or down as needed.
And the list just keeps growing. Scientists have found proteins that act as an off switch for CRISPR, giving us more safety and control. They're attaching tiny fluorescent lights to it, not to edit, but to literally watch where specific genes are inside a living cell in real time. They're even using it to run massive experiments, knocking out thousands of genes at once to quickly figure out what each one does. The system is just incredibly versatile.
And all of this brings us to our final and maybe most important section. The future is editable. This isn't just theory anymore. CRISPR is actively writing the next chapter of science and medicine right now.
And you can already see its impact everywhere. A huge chunk is in biomedical research, where it's letting us understand diseases faster than ever before. It's also making major waves in human therapeutics, with clinical trials underway for genetic conditions. And beyond that, it's changing agriculture, it's helping us create cleaner biofuels, it's really touching almost every part of our world.
And this quote from our source material just nails it. It is not an overstatement to say that CRISPR has completely redefined the entire field of genome engineering. It blew the doors wide open on possibilities that were just locked shut a little over a decade ago. So, we'll leave you with this thought. For the very first time in human history, we hold the tool to rewrite the biological world around us, and even within us. The big question is no longer, can we do it? The question is, what should we write? It's a huge responsibility and one we all need to start thinking about. Thanks for watching.